Welcome to the PubMed Daily Review for May 11th, 2026. Today we scanned 1286 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, diverse surgical and clinical cases. This trend examines diverse, complex surgical and clinical cases, highlighting diagnostic dilemmas and evolving treatment approaches. This cluster covers 305 papers. Representative work includes neo-left colon volvulus or metachronous descending colon volvulus following sigmoidectomy for sigmoid volvulus, laparoscopic resection of a colovesical fistula secondary to diverticulitis using near-infrared fluorescent ureteral catheters, and challenges in surgical training in the United Kingdom and potential solutions. A narrative review. Trend 2 of 5, comprehensive cancer research. This trend investigates cancer across its entire spectrum, including biology, diagnosis, treatment, epidemiology, and patient care. This cluster covers 174 papers. Representative work includes tumor-infiltrating lymphocyte therapy for melanoma and other solid tumors. Looking back, yet moving forward, temporal trends of colorectal cancer burden in India, a nationwide and state-level analysis, and microsurgical oncotesticular sperm extraction in a patient with testicular cancer and congenital unilateral testicular absence. A case report. Trend 3 of 5, basic biological and ecological research, unraveling fundamental biological processes and ecological interactions across diverse organisms and their environments. This cluster covers 388 papers. Representative work includes field-based mesocosm evidence for the role of natural nutrient sources in cyanobacterial blooms, growth rates, and microcystin production of microcystis strains in response to allochthonous and autochthonous nutrient sources, and evaluation of Arabica coffee pulp extract on sperm quality and hematology in cyclophosphamide-treated rats. Trend 4 of 5, public health and epidemiology. This trend investigates diverse health determinants, disparities, and interventions across populations using epidemiological and public health methodologies. This cluster covers 238 papers. Representative work includes environmental monitoring and smoking exposure before and after the designation of non-smoking areas in indoor sport facilities, characterizing infectious disease mortality in severe mental illness. A retrospective match cohort study, and time windows used when identifying current drug use and polypharmacy. Trend 5 of 5, psychosis and cognitive disorders. Research explores biological mechanisms, cognitive deficits, and treatment strategies for psychosis and related cognitive disorders. This cluster covers 181 papers. Representative work includes membrane interfacial potential governs surface condensation and fibrillation of alpha-synuclein in neurons, passage of time or delay signal at the level of milliseconds, a new approach and a selective difficulty in individuals with schizophrenia, and tracking symptom change across therapy sessions in virtual reality cognitive behavioral therapy versus cognitive behavioral therapy for paranoid ideation. A secondary analysis of a randomized control trial. Up next, our first paper out of 5, Dissecting Human Embryogenesis with Blastoids. Insights into Lineage, Specification, and Early Development by Hiro Sasaki and Ayaki Yanagita from Department of Veterinary Anatomy, the University of Tokyo, Tokyo, Japan. Let's break down its key contributions. Understanding how human life begins is a fundamental scientific challenge, but studying early human embryos directly is incredibly difficult due to ethical and technical hurdles. This paper explores a groundbreaking solution: blastoids. These stem cell-based models offer a new window into the earliest stages of human development, which is vital for improving fertility treatments and understanding the origins of birth defects. This paper is a comprehensive review, synthesizing existing literature on human stem cell-based embryo models, specifically blastoids. Researchers systematically searched PubMed to gather studies on how blastoids are generated, their diverse forms, and the insights they provided into early human development. The fidelity of these models was continuously compared against the known processes of natural human embryogenesis. The review highlighted that blastoids successfully model key aspects of blastocyst development, including lineage specification and early morphogenesis. They've proven incredibly useful for studying complex processes like implantation, embryo-maternal interactions, and responses to various perturbations. However, the review also pointed out current limitations, such as incomplete lineage representation and restricted post-implantation modeling. Excitingly, ongoing advances in stem cell biology are expected to significantly improve their fidelity and developmental potential. This review underscores the profound clinical significance of blastoids. They provide an ethical and technically feasible alternative to studying human embryos directly, pushing forward our understanding of developmental biology and reproductive medicine. While current models have limitations, such as incomplete lineage representation, these are actively being addressed. Continued research and technological advancements are vital to refine blastoids, unlocking their full potential to unravel the mysteries of human development and disease. Up next, our second paper out of 5, biopsy with same-session MRI-guided laser interstitial thermal therapy versus biopsy alone in patients with primary unresectable glioblastoma. A multicentre randomized controlled trial by Celine L.G. Noodle and Mark Terlaan from A Department of Neurosurgery, Radboud University Medical Center, Nijmegen, the Netherlands. Let's break down its key contributions. Glioblastoma is a devastating brain cancer, and for many patients, surgical removal is an option. This leaves them with limited treatment choices and a very poor prognosis. Laser interstitial thermal therapy, or LIT, offers a promising, minimally invasive approach to treat these tumors. However, before this study, we lacked robust evidence to truly understand its benefits in this challenging patient population. This was a multi-center randomized controlled trial, the EMIT trial, conducted across 7 Dutch hospitals. We enrolled 26 adult patients diagnosed with primary unresectable glioblastoma. Patients were randomly assigned to either receive a biopsy followed by standard care or a biopsy combined with same-session MRI-guided LIT, also followed by standard care. Our primary goals were to assess overall survival and health-related quality of life at 5 months post-randomization. Now, let's look at the numbers. The median overall survival in the LIT group was 7.8 months, which was notably longer than the 4.8 months observed in the control group. When it came to health-related quality of life, both groups had comparable mean QLQ, C30 scores at 5 months, suggesting LIT didn't negatively impact patient well-being. However, it's crucial to remember that this trial was terminated early due to slow patient recruitment, meaning our sample size was smaller than planned. So, what does this mean for patients? While we can't draw definitive conclusions due to the trial's early termination and small sample size, these results suggest LIT holds promise for extending survival in patients with unresectable glioblastoma without compromising their quality of life. This study highlights the critical need for larger, well-designed trials to confirm these findings. The lessons learned from the EMIT trial will be invaluable in shaping future research efforts to bring this potential treatment to more patients. Up next, our third paper out of 5, an inflammation-targeted lipid nanoparticle inhibiting ferroptosis for spinal cord injury repair by Yao L.V. and Min Ge from A Department of Neurosurgery, Suzhou Affiliated Hospital of Wenzhou Medical University, Suzhou People's Hospital, Suzhou, 324000, China. Let's break down its key contributions. Spinal cord injury is a devastating condition, leading to severe and lasting disability. A major challenge in treatment is the secondary injury cascade, involving inflammation and a specific type of cell death called ferroptosis. Erythropoietin, or EPO, has shown great potential in preclinical studies to protect the nervous system. However, delivering EPO effectively to the injured spinal cord without causing harmful side effects has been a significant hurdle in clinical trials. This paper explores a novel approach to overcome these delivery challenges. To tackle this, researchers engineered mannose-modified lipid nanoparticles, or MLNPs, to specifically deliver EPO mRNA to the injury site. They tested these nanoparticles in both cell cultures and a mouse model of spinal cord injury. Their methods included advanced imaging techniques like bioluminescence, detailed cellular staining, and molecular analyses, such as Western blotting and ELISA, to track the nanoparticles and evaluate their effects on inflammation and cell death pathways. The results were very encouraging. The targeted MLNPs showed significantly higher accumulation and expression of EPO mRNA at the spinal cord injury site compared to untargeted nanoparticles. This led to a marked downregulation of key pro-inflammatory cytokines like TNF-alpha, IL-6, and IL-1 beta. Crucially, the treatment also significantly increased the number of surviving neurons and preserved axonal integrity, ultimately resulting in a marked improvement in motor functional recovery in the SCI mouse model. Mechanistically, the study confirmed that this therapeutic effect was partly due to the suppression of ferroptosis, a specific type of cell death, by regulating iron metabolism. This study presents a significant step forward, demonstrating that targeted delivery of EPO mRNA via these smart nanoparticles can overcome the long-standing challenges of systemic EPO administration for spinal cord injury. By simultaneously tackling both inflammation and ferroptosis, this approach offers a powerful new therapeutic strategy. While these are exciting preclinical results, further research is needed to translate these findings into clinical practice. However, this modular nanoparticle design holds great promise not just for SCI, but potentially for other neuroinflammatory disorders as well. Up next, our fourth paper out of 5, Secure Bioinformatics, Privacy-Preserving Federated Analytics Using Homomorphic Encryption by Weizhuang Zhou and Qinmi Miang. Let's break down its key contributions. Imagine the challenge of analyzing vast amounts of sensitive genetic data spread across different institutions. Current methods often require moving all that data to one place, which is slow, hard to scale, and a huge privacy risk. This paper tackles that very problem, asking how we can collaborate on these analyses securely without ever centralizing the raw data. The researchers developed a novel framework for secure bioinformatics. Instead of moving sensitive data, they used privacy-preserving federated analytics powered by homomorphic encryption. This innovative technique allows computations to be performed directly on encrypted data, ensuring privacy while still yielding accurate results. They validated this approach using real-world cohorts. The results were impressive. The framework achieved over 99.9% accuracy compared to traditional plaintext analyses, showing that privacy doesn't have to come at the cost of precision. Importantly, it also maintains scalable runtime performance, even with increasing data size and more participating sites. This clearly demonstrates the practical feasibility of secure federated analytics for sensitive bioinformatics applications. This research has significant implications. It provides a robust solution for secure, large-scale bioinformatics collaborations, eliminating the need to transfer sensitive raw data. By addressing critical privacy concerns, it paves the way for broader data sharing and accelerates discovery in genomics and other biomedical fields. This framework could truly revolutionize how we conduct collaborative research with sensitive information. Up next, our fifth paper out of 5, Biallelic Germline MBD, 4 mutations predispose to colorectal polyposis, hypermutated AML, and schwannomas by Julia Cooper and James S. Blockley from Human Genetics, Department of Internal Medicine, The Ohio State University, Columbus, Ohio, USA. Let's break down its key contributions. Today, we're diving into a fascinating case series that sheds new light on a rare genetic condition called MBD4, associated neoplasia syndrome, or MANS. This syndrome is caused by mutations in the MBD4 gene, and while we know it predisposes individuals to early-onset colorectal polyps and a specific type of leukemia, the full range of its effect has been less clear. Understanding this broader picture is absolutely vital for diagnosing patients earlier and managing their care more effectively. This study is a case series, focusing on 4 individuals from 3 different families, all diagnosed with MANS due to biallelic MBD4 mutations. The researchers meticulously documented their clinical histories to understand the diverse health issues these patients faced. As a case series, there wasn't a comparator group or a specific intervention. The goal was to observe and characterize the syndrome's manifestations in detail. The primary outcome was to define the full range of cancers and other conditions linked to MANS, observed over their clinical course at 2 specialized centers. The study identified 4 new patients with MANS, increasing the total reported cases by 27%. All patients developed AML between ages 28 and 39, and all had early onset adenomatous polyposis. Importantly, the researchers discovered new associations. 2 patients developed schwannomas, and 1 developed thyroid cancer. Genomic sequencing of these tumors revealed a striking finding: a very high tumor mutational burden, sometimes exceeding the 97th percentile, confirming the hypermutated signature driven by MBD4 deficiency. This research significantly broadens our understanding of MANS, showing it's not just about polyps and leukemia, but can also involve schwannomas and thyroid cancer. This means we need to be more vigilant in screening for MBD4 mutations, especially in patients with unexplained early-onset cancers. Moving forward, clinical guidelines should consider expanding surveillance to include the nervous system and thyroid. Furthermore, the high mutational burden observed in these tumors suggests a potential for new therapeutic avenues, such as immune checkpoint blockade, offering hope for future treatments. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.